Hello. Good afternoon. So our next speaker is Professor Sebastian Oslin. And Professor Oslin is the head of the School of Biomedical Engineering and Imaging Sciences at King's College London. He is also the director of the Wellcome EPSRC Center for Medical Engineering and also deputy director of the London Medical and Imaging and Artificial Intelligence Center for Value-Based Healthcare. And today he's going to talk to us about learning across 16 million patients, platforms and applications in acute care.
Thank you very much. If you could project the slides, that would be helpful. Great. Well, thank you very much, and good afternoon, ladies and gentlemen. I must say that I'm amazed by the amount of people in the audience, given the amazing weather you've got out there. So I'm really grateful that you're going to bear with me for the next 30 minutes. So I'm Seb Oslin. I've been working in machine learning and medical devices for almost 25 years. And over the last 5 years, we've been working quite hard with many partners to build a platform which hopefully can take advantage of all of those hidden data sets which are within our different hospitals across the National Health Service, NHS in the UK. Just to put things into perspective, I just wanted to start by perhaps repeating what has been already said in terms of the challenge of the healthcare model. And the model I'm showing here is a very common model across Europe and North America, where patients or cost related to patient care is a very simple equation, which is basically just the addition of how many patients you have treated, what are the numbers of procedures, what is the reimbursement fee associated with them, and this gives you the healthcare cost. And unfortunately, this model, with the current population as well aging, which costs roughly 30% more than patients who are below the year of 50, is actually unsustainable. And what I'm showing you here is, in fact, the medical care price growth versus the care of goods or price of goods, usually. And you can see the huge delta between how fast the medical care grows versus the consumer prices. And those numbers continue to diverge from one to each other. And if one will follow, just in the UK environment, if one will follow the medical care prices rather than the consumer prices, all of your goods could be roughly double of the price that you have right now. And this is not only true for the cost itself, but as well from a percentage of GDP. And unfortunately, what you see here, and I don't have Italy here, so I can't be blamed that I've got numbers wrong from your country, is basically the healthcare spending as a percentage of GDP. And those numbers are quite staggering. And what you can see, for instance, for the US, we're talking about over 16.5% of the GDP, which is consumed every year just only to provide care. And this numbers is always growing. In a country like the UK, the numbers have been basically blocked at roughly 10.5%, 11%. Not because we need to spend more, because we do, but we cannot afford it. And if you look at this from a consumer point of view, if you spend a lot of your tax money, I'm talking about people tax here, for your care, you will expect a good service. And especially if you spend so much, if every single of your dollar or your pound or your euro, 10 cents of it is going straight to your care, one will expect to be satisfied with their care. But if you look at the satisfaction of care, this is pretty grim. The worst is actually the one who spends the most, which is the US, which is really at the bottom of patient satisfaction. And the one from Europe here are all actually, frankly, pretty average in terms of satisfaction. So clearly the model is completely dysfunctional, not only from the cost point of view, but for the satisfaction point of view. And this requires to think about the healthcare model in a completely different way. And this has been really pushed over the last 10 years. It's very hard to implement, and it's a concept of value-based healthcare. So I just remind you the model, how it looks like, and clearly the patient is seen as an individual, not across a pathway, but within a specific care. So if you, for instance, need to go to the hospital to identify if you've got, unfortunately, a cancer, you might actually do a scan, and then you go to radiology, and you will be costed versus radiology activity. Then you might have to do a biopsy, and then you might have actually eventually to go to oncology. The model really that hopefully we could all reach, which is not easy to implement, it's a value-based healthcare model. When rather than looking at a fee for service, you look at the fee for outcomes. And so you look at the patient around the entire pathway, from eventually at some point prevention, if we can implement it, which is certainly very challenging from a financial point of view, up to post-assessment after intervention. And then the health outcome is going to be very, very different. But if you want to do that, if you want to implement this, you need to have access to the data of those patients. And at the moment, those data are clearly blocked in the different care, from primary to secondary to tertiary care, and are very hard to link together. And that was really the foundation of the rationale behind the vision of Healthcare 4.0. And Healthcare 4.0 is something we would like to embrace. Everyone, in fact, working right now in AI and digital health is focusing towards access to those data to be able to optimize the value out, or the quality and the impact out of any care in any healthcare system, with the caveat that it works much better in a single payer model for value-based healthcare than in a private model. But really our desire and the vision of our center has been really to see how we can use imaging, data science, and AI to deliver value-based healthcare. So we've been focusing mostly on radiology AI, because that's where we have the most expertise, but most of what we discuss here could be eventually translated into other areas. So this was our statement at the time we created our center, which was in 2019. And I just want to put things into perspective to the fact that we are now in 2024, and this is what Eric Topol wrote in 2018. And in 2018, he was making the claim that all of those data are just sitting there, and now they have been analyzed and interpreted, and it is the most radical change happening in healthcare. I just want you to reflect on the fact this was written in 2018. I've got massive respect for Professor Topol. He's clearly a key opinion leader, but I think if I was just changing 2018 by 2024, you will still believe it. And the reason is we're still working with the future tense. So the amount of AI application in healthcare is irrelevant right now in any hospital setting. Right? There is a big challenge of getting those technology into care for patients. And this challenge is even more prominent post-pandemic because the systems is not able financially to even support the replacement of some of the very old IT infrastructures that most of our hospitals have. So what we decided to do was to identify what was really the key pinch point, and they were not going back to the presentation from our colleague about the multidisciplinarity aspect. Those pinch points were not only about AI and data expertise. They were about transforming pathway in the hospital. They were about access to data. They were about partnership across not only discipline, but sectors, public-private partnership. They were about public and patient engagements, regulatory approval. They were about governance, safety. They were about health economics and reimbursement, and it's still a very difficult topic, reimbursement in digital health. Clearly there is an issue. This concept of perceptive value. If I ask you how much costs a car, I'm sure you've got an idea. If I ask you how much costs a snack bar, I'm sure you will not tell me 20,000 euro. But if I ask you how much costs this computer-aided diagnosis tool to implement into your healthcare system, you will have as many answers than people in this room. And this is really a big challenge that we are facing. And of course, there is a challenge of the hospital data access infrastructure, which by itself can be seen as a mode to protect our patient data. And so how do you penetrate this and how you make it happen? So we built a consortium. We started, as I said, in 2019. We started with 3 universities and 4 NHS trusts. We embed within our consortiums many patient pathways, and then we involve startups and multinationals to try to really understand how we can better partner with those companies, how we can better validate some of their technology. And then from there, we actually expanded from a proof of concept to a very large scale of what was 10, which became 11 trusts now that the Birmingham NHS Trust has joined us, to really professionalize the phase 1 proof of concept that we developed, which I will present to you, to hopefully create what could become a standard of what we called an AI-enabled hospital, which gives you access to data interoperability, which protect patient privacy, which is scalable and has a very strong data model beyond it, following standards of the lack of OMOPs, which has an appropriate computational facility with a mix of on-site and on-cloud, and hopefully have an appropriate data governance, which enable you to get access to data. And access to data, it's still the same issue today than it was 5 years ago, and I think we need to appreciate that. So what is the difference between doing some interesting experiments in your labs when you have the data set and doing it into a hospital? It's all this concept of static snapshot versus real-time actionable analytics. And if you try today to implement anything in an hospital, those timing I'm putting here are really true stories of the time it takes for people to get access to data, to interpret the results, to get this data back. And clearly this is not by any means, shape or form, real time. And if you really talk about actionable AI delivered in an hospital, this needs to happen in a matter of seconds, not in a matter of years. And so you really have to really rethink the whole system. And one of the parts of the systems that we've been working on is what is the appropriate ethics and governance. I'm not going to the great detail of this, but we spent nearly 3 years to actually get this in place, building a full information governance, a data allocation committee, which includes senior leadership of those different NHS trusts. We build it with privacy by design, using federated learning, which I will mention later. We had to have appropriate transparency and best practice, and very, very importantly, we had to have a robust patient and public involvement on a project basis. Because clearly, the big challenge we have as well as member of the public and the community is trying to explain and having this conversation that if you really want to make a difference to patients with digital health, you need to build products. Why? Because this is a very heavily regulated environment. It's not about building a proof of concept. It's about building a technology. And this technology needs to follow quality management system, ISO 13485, being appropriately approved by the European community and so on, go to FDA. And to do that, you need to have an enterprise beyond you. And that's really a challenge of a conversation. You will get access to my data to build a product, what is in for me. And it's coming back as well to the reimbursement model, which I think still needs to be challenged. And the other challenge is we're talking about real data, clinical data. And the clinical data have different shape or form. There is missing data. There is causality in those data. They are dirty. They are unstructured. Quite often, those data are not aggregated properly. Even if you've got an electronic health record of the lack of Cerner or Epic, you will still have other databases which are not linked, including your PACS, which somehow has a connectivity with it, but will be seen as an independent system. They will not be structures, all of them, but some of them will be. And how do you link with all of that? So we had to build really a full infrastructure and spend a fair amount of money. We're talking about tens of millions of pounds, which has been invested into this. Have a partnership between key industries, and some of them now are very well known of the lack of Run AI. We were one of the first, if not the first, in the UK to use it. Work very closely with NVIDIA on some of the compute. Work closely with open source frameworks to make sure that the community as well can take advantage of some of the development we've been doing. And really changing the model as well in terms of the framework related to moving your models rather than moving your data, which is really at the basis of, at the root of federated learning. So very, very briefly, because I clearly don't have time, we had to build a system which enabled us to build what we call a secure data enclave in every single of those hospitals, and to gather those data eventually together if we wish to. Again, we use open source standards that we contribute to the development of the lack of XNAT, but as well CockStack, which is based on natural language processing, and an engine called Elastics, which as well is open source. And this combination is really, really critical. Quite quite often people are looking at the data from the radiology packs independently from the e-noting or any type of radiology reporting. But if you look at radiology reports, this is really where you have massive challenges, and I'm just giving you one here. For some reason, and I'm sure I've got a lot of radiologists in this room, radiologists love to use double negation. They like to say what is not appearing sometimes as much as what is there. And so if you just look at this independently without understanding the semantic, then you will very likely convert what was a diagnosis against what was a counter-indication, for instance, or counter-diagnosis. So you have spellings, you have typo, you have issue of acronyms, you've got double negations, you've got all the family story which might be mentioned, which says that your mom might have a breast cancer, but you're male. So now you've got the word breast cancer in this report. Does it mean you have breast cancer yourself? And all of this needs to be considered. And it can be done. And we build as well another software called Semantic EHR. It's online. You can use it. Integrate it into a hospital framework. Here I'm giving you one specific example. When you see on the right a very nice chart, which was a plot from a very large US study, which involved hundreds of people collecting data over many years. It costed millions of dollars actually to build, just to look at the prevalence and the use of certain medications along the year, specifically against stroke. And what you see on the left-hand side, which still looks a bit dirty, but very similar, if you forgive me for the color map, very similar to what you see on the right-hand side, is a fully automated system which actually just took an afternoon to build of the same type of data set along the year from every single patient who actually received some of those drugs over the last 10 years at King's College Hospital. So clearly, if you can really get those semantics in place, there is amazing information that can be extracted. And again, I'd like to just make an highlight of the OMOP standard, which I think is really, really important. And if we were all using it, I think we could start to share our structured data sets. And then there is, of course, what do you do with those data in terms of AI? And so we worked very closely with NVIDIA a few years ago to start a consortium named MONAI, which by now I think has become quite a key platform, open source for our community, especially in medical imaging. It has been downloaded now, has been told, over 2 million times. So I think people have been embracing it. And it enables us to be able to compare our results without having all the noise about I/O or any type of ways that you've been building your model. And then if you have now this whole framework together, then you can tackle actionable models. You can build those models. You can tackle precision medicine. You can do business intelligence. It's not all about improving patients through care. It can be improving patients through workflow management, through patient waiting list, through automatic referrals, through follow-up. So the non-clinical pathway in an hospital is as important than the clinical pathway. And I think it's been under-researched by your community. So once we've got this in place, the question is really how do you go beyond the firewall of your hospital? And this is really where the whole concept of federated learning is coming. Again, there is many ways of looking at federated learning, but in a nutshell, it's really about how can you connect those data sets without having to move any data, which really enhance your privacy preserving. Of course, it's going to be always challenging to get the best model, which means that if you have a lake, then of course your model might be better than having those data trained or those model trained by just moving around the weight of your gradients optimization. But fundamentally, it will always be better than only one single site. And we've been actually getting the first ethics across the UK to be able to do federated learning across those now, what is 11 trusts. And then once you are able to build your model and to train it at scale with this amount of data, what you need to do now is to deploy it, and to deploy it safely. And that's really the concept of building what we call an AI deployment engine. And so making sure that you are vendor neutral, making sure that you are DICOM compliant, making sure that you can include as well your non-imaging data. And we decided to partner with another company called DeepSea, which is based in Munich and now has a subsidiary in the UK, which is in fact within our own hospital, to really tackle those issues of being FHR compliant, to be able to integrate non-imaging data, to have a single central repository, to be able to build this as an AI evaluation framework, to build a sandbox, a safe sandbox, to be able to validate your model which are not CE mark. So the whole infrastructures that you need to be able to deploy radiology safely. And now I would like to spend perhaps the next 10 minutes just to give you a bit of a sense about what could be achieved by following some of those models. And most of the models I'm showing are now part of this Monai Gen AI, a generative AI platform, which we release open source as part of Monai. And we've been tackling specifically AI-enabled neurology. And if you've been looking at the recent Lancet review, neurodegenerative disease, any type of neuro disease, neurodisability is now the largest burden that we see in Europe and North America. So it's a very, very key healthcare problem to tackle. So we wanted to be able to see how we could use stroke as a proof of concept to demonstrate how we could scale up our AI model, how we can use all this data, which are highly multimodal, within a hospital setting. It is really a big data problem. Here we're talking about over 2 million volumes. Here we selected 150,000 patients as a first proof of concept within our hospital settings. Just to give you an idea, 150,000 patients, which means that in terms of MR sequences, 14,000 different sequence parameters, 14,000. So if you think about standards, you take a picture, you think there is one way of doing an autofocus on your camera. Here you've got 14,000 parameters. That means that every single image, or 10 images only have the same parameters, which is quite outstanding. You've got a mix of field strengths as well from your MR scanner, 1.5T, 3T. We even had data on some of our other studies of lower field or higher field. You've got your risk report, and then you've got your ICD-10 code, which is really important if you really want to link it with a specific disease pathway. And there is another issue when you look at research data versus real-world data. Those are the type of data sets that you will get in the UK Biobank. They look lovely, amazing. It's a nice brain, it's a healthy brain, and it's very good resolution. Those are the standard data you will get within the NHS environment. You are limited in terms of time. You are limited in terms of cost. So therefore, you will get whatever is available as a machine. You're not going to be able to provide a standard operating procedures about the protocol. You will do what you've been told as a radiographer, frankly, and just scan in the time-limited fashion. So the data are really an isotope. The other issue, again, is sometimes DICOM doesn't tell you everything. It has been manually entered. It could have been mistyped. But more importantly as well, some data might be labeled as a brain and might not be a brain. Some data have major artifact, ghosting artifact. Some data might actually miss half of the brain in this case, or it actually be a neck. Or some data are extremely pathological. And I'm showing you a real case of a patient who had a full hemisphere removal. And of course, the other challenge as well is you've got missing data, different resolution. You've got as well different of scanners. You've got different type of artifacts. But very importantly, if you look at it across different hospitals, for the same pathway, you might have as many different contrasts coming from your scanners. And then you try to actually make sense of it, and you try to measure, let's say in this case, those plaques, you try to measure it consistently, and that's really where this concept of generability is really important in your AI model. How can you make sure that you're not specific or biased by one specific scanner? So where do we start, and how do we ensure robustness? So let's look first at those annotations. So we took those 150,000 patients. We did a full report labeling, and once it's done, we were able to do clustering. And this figure shows over 150,000 patients. So it's a hierarchical cluster of the radiology report, using, in this case, a RADLEX cluster. And what is quite fascinating is those specific colors. They represent the same high-level concept that we will have identified from those reports. It can be dementia, it can be stroke, and so on. And why this is powerful? Well, first, it helps you to make sure that you've got a right representation of your patient population. You can identify the healthy the super healthy as well. You can identify those different subgroups, and what you can identify is the inhomogeneity. So if we build a model, and you want to represent all of the disease, you have to have an even representation of all of your different disease states, right? That's really important. And something really key for us is we want to identify the control. We want to identify the healthy patient, because we want to be able to build a model which will tell us how we are what we call out of a distribution of normality. And this concept of out of distribution is really, really important in the context of latent models, which became extremely popular with diffusion models. So we started to look at what was called, or still called, variational autoencoder, which became extremely popular in the early 2020, when we had some outstanding work at the time by Bo et al., which really started to introduce into medical imaging, some of the concept of in and out distribution detection, how you can do abnormality detections, in this case using transformers, which became extremely popular. But I think it's important to remind everyone that transformer didn't start when OpenAI launched. In 2017, we had seminal papers in transformers, and it's been used for many years by the NLP community. And so the whole point of this is to be able to generate data. And generating data is not only important for the sake of increasing the size of your database or enhancing patient privacy by creating data which are synthetics, very representative of your population at a very large scale, which can eventually be used for either in silico clinical trials or making sure that you've got enough data to build a robust model. But as well, fundamentally, they will be extremely useful to identify what is normal versus what is abnormal. And that's really the starting point of many of the technologies that we've been developing. So we use a concept of what is called VQVA, Vector Quantized Variational Autoencoder, which is a mouthful way of just saying you use transformers here to do your quantization. That works pretty well. It has still a lot of challenge, especially in terms of scale. So you will actually be able to synthesize images, but you will have a challenge in terms of going to higher resolution. But you still can build pretty nice brains. So here, and I'm giving you the website when you can generate your own brain if you want. This is a fully generated brain, which was based on 33,000 samples from the UK Biobank. It was based on a VQVA. It was using L2 loss, but I'm not going to go into detail on this, using roughly 10,000 tokens, because it's limited in terms of what we can do and how we can fit transformers to build in the memory of GPUs to build 3D brains. And going from 2D to 3D is hard. It's a very hard problem. I think it still looks like a pretty convincing brain. But then if you move from transformers to what we call LDM, latent diffusion model, then you can really break the barrier of resolution. You can go much further. And so you change your quantization here, and rather than using a single vector which is long, and the trouble is how long it can be, you actually use convolutions in your diffusion model. Then you can have a much, much larger quantizations. And then more importantly, you can move from what was 32, 64 cubic images to actually build that full resolution your image, 256 cube. And then you can generate those type of image, which I think are quite staggering. So if I was asking you which one is fake, which one is real, does anyone think the one on the top is fake? Don't be shy. So in this case, the bottom one is fake. So we did a study with more than a dozen of our radiologists, experienced radiologists. We gave them only 10 seconds to look at those images. We had random sample at the end. We had 50-50 between accurate versus fake, right? Or real versus fake. If you have more than 10 seconds and you look at carefully at every single slide, you will identify at some point some issue. But it's not as far off to what people can actually believe this is real. And what is quite outstanding as well is you can create those type of images. It's very dark. It's not meant to be that dark. But this was to show you that we can even create and generate lacunes into the brain. You can really go to very high resolution. And what is really exciting, you can even build models in 4D. And in this case, we generate an image with large ventricles or small and large brain volumes as a proportion of your total intracranial volumes. And we can actually generate even an image over time of the same patient, which again can be extremely helpful if you want to build a model of degeneration. You can use those latent models to identify, as I say, what is in the distribution versus what is out. Why this is important? Because again, if you cannot trust the content of your DICOM tags, how do you know you're looking at a brain when you have to have 20 million brains? You're not going to look at them manually. So we build models which enable us to actually assess with 100% accuracy that this model is looking at a brain image. We can as well look at very subtle change, what we call near out of distribution, when you've got some artifact, like a missing slab within your MR scan. You can then look at how to detect outliers using those models, and that's giving you, for instance, in this model, built on over 100,000 of healthy brain, automatically identified something that has never been seen by the model. In this case, it could be plaque, it could be a stroke, or it could be a tumor. And then because you've got this latent diffusion model, you can actually as well model uncertainty. So rather than use model basically how unsafe your model is. I mean, you always want to be in the ideal situation. You want to be very certain, and you're correct that you're very certain. So worst case scenario is you are absolutely certain and you're completely wrong, right? And this concept of uncertainty has not been really well captured in most of the model, and quite often the model we have in our community are very confident to be right while they are totally wrong. And so if you start to use uncertainty as a way of removing data set from your samples to only focus on the one you've got high certainty, then this becomes a really big challenge. Don't want to go into much detail, but just to give you an idea on one example, here we were trying to segment specifically the stroke in this image, and what you can see is there is very, very high uncertainty around area which are edema, which are outside of the brain, which is exactly what we want to have. One of the other challenges that you see in medical imaging that you don't see in natural image is the field of view is different, the resolution is different. So you end up in a situation that you are limited in terms of the data you can collect and you can use together. So in this case, whole body scan. And what we did is build models when you can include the geometry and a grid conditioning into your model. And what is remarkable now is from a whole body, you can generate any image, half of an image, upper body, lower body. But more importantly, those models become having an emergent behavior of super resolution, which enable you to actually build scan at a higher resolution than the one you had, and we demonstrated actually the value of this from a point of view of super resolution. I'm going to skip those parts because I've been told that I'm late, but I just wanted to highlight one part, which is around the MR physics, and to give you a sense as well. So when we're talking about those volumes, here I'm showing you 4 scans from an Achiever, a Prisma, a GE system, and a Scara, another Siemens system, when you've got basically different volume measurement from your scanner in terms of white matter measurement or total intracranial volumes. So what we did is went to the next step and start to model the physics of the MR acquisition into your generative models. So not only trusting the image, but adding into your image a full simulation using simulation block, which is a very well-known models to be able to create what we call multi-parametric MR, MPM. And not only this improve your model, but it make it insensitive to those different parameters I was talking about. And then you can have actually repetitive measurement while you can change, for instance, the inversion time on your scanner. And we increase this up to the point of now being able to make it multi-modals, being able to go to super resolutions on a mix of T1, T2, and FLAIR. And all of those type of algorithms were now embedded all together into one single step. And in this case, it was for building automatically a report for stroke, which has many steps. I'm not going to go into detail, but end up by bringing into the packs a full PDF form, which can be open, using all of those different algorithms to end up by having a full phenotype and reporting for this specific patient. So I'm sorry I had to rush a bit at the end, but in conclusion, I think retrospective data which are sitting there in the hospital have immense value. It's very hard to extract them so far, and we believe we have a framework to do that, not only from an algorithmic point of view, but as well from a data privacy and a governance point of view. we believe that it is helping to create some of those standards for an AI-enabled hospital, and very importantly, create an ecosystem for innovator and our tech transfer. So now we've got more than 20 companies working next to us, physically co-located with the hospital environment, which can safely test those algorithms. And last point is we are actually already expanding internationally, having some formal collaboration with France and in the US now, and we'll be really excited to see if there is any opportunity to link with our colleagues in Italy. Just want to thank you for your attention and very happy to take any questions.
So we have time to take maybe one question.
Thanks, Sebastian. That's great. So here in Padova, there is a conversation to build a new hospital. That should be the hospital of the future. I see your presentations. I see the future is already here. So do you have any suggestions to the policymaker, to those involved in the building this new hospital, to how to copy this winning model?
Well, thanks for the very targeted question. So to put things into perspective, I lead a school which is fully embedded into an hospital. We are more than 700 people, 88 academics, all working under the same department, while we have surgeons, physicians, radiotherapists, chemists, biologists, computer scientists, roboticists. So it completely breaks the mold of how university meant to look like, which is very pillar-based. So all of those people are under one umbrella, which is the School of Biomedical Engineering and Imaging Sciences. So I think the co-location is key for the future of healthcare. Most of the revolution, I'm sorry to say to all of our life scientists, but the revolution is going to come from computer scientists, come from medtech people, come from pharma AI solutions. So there's a lot of revolution going to happen from computer scientists, and it needs to be validated properly. And if we just do that outside of the clinical environment, we're going to waste the next decade just to try to bring those models with this type of skill into the hospital. So the first thing I will do, if I was building a new hospital, is I will make sure I've got enough space to embed all these multidisciplinary schemes, and it might as well be some of them on social sciences. You know, we're talking about philosophers. You know, we've got colleagues who are actually from philosophy within the framework of our digital health hub, who are as well from the social science context, who are actually from law as well. How can we embed that? I mean, if you've been doing any ethics or writing any research programs and try to do any clinical trial, I can guarantee you spend a lot of time to make sure that you are compliant with a lot of laws. And having this embedded with your team is really important. I will make sure that the university get more involved into providing cares in a safe way. So being able to mix the research on your devices with the clinical delivery. Yeah, easy.
Thank you very much, and I guess there will be more questions during the coffee break. Thanks. Moving on to session 6 directly on AI applications. That will be the first part. And let us invite Professor Henning Müller. He's professor at the HCS SO Valais. Since 2011, he is responsible for the health unit of the school. And since 2014, he's a professor at the medical faculty of the University of Geneva. And finally, since 2020, he is also a member of the Swiss National Research Council. So welcome, the floor is yours.
So thank you for the introduction. Thanks also for inviting me. So I'm really happy to present some of the results of our research projects here. It's a project called ExaMode, EU-funded project where also the University of Padova participates. So it'll be work that I'll present that is from a variety of backgrounds, and many of the things have been said in some of the presentations before already. So it's interesting to see how things overlap, are sometimes different, and also come often to very similar conclusions. So it's the project is called ExaMode, and histopathology data analysis, and I think I have a laser pointer here. Well, And what you can see here on the slide is basically the outcome. And I will explain you how we got to that, how we used like image data together with text reports to basically fully automatically train the models like without human interventions and annotations. And currently when I talk to people about AI, often it's like, oh, ChatGPT and whatever will happen and how it will change the way we work and kill the workforce. And I took one video, it's on YouTube, so you can search for that. It's a deep learning conference in 2016 where people actually predicted within 5 years there will be no radiologists anymore, and so we should stop training them. Hasn't really happened, and I mean, we've seen that before in a couple of presentations. Things change much, much slower than we often think, and there's a lot of problems that were presented in the talk just before. I mean, there's so many challenges in medical data when you want to bring it really to the clinic. I mean, you have so many different machines, you have changing machines, so you're hitting on a moving target. And I think all these things need to be taken into account. And I've just randomly put up a few of these, like, so machines are better, and well, you can beat doctors and outperforming, but then whenever you put it into a real clinical practice, often it just doesn't work as it works in the lab. And I mean, what we really wanted to do here with in the XR mode project is do a very practical approach and look at some of the things. I mean, also, we need to take into account what the patients think about it. And currently, they don't really trust the AI algorithms. And so we need to look at like what, we need to have people who interact with the patients as well, explain the decisions, and all this interaction is really important. It's not only about like the technology. But when we started the project, I mean, there were a couple of challenges that we had identified. We have a very large amount of data. Much of it is not necessarily easily accessible, so we need to look into what we can use, what we can't use. Annotations are very expensive, so we wanted to work with weak annotations and how we can also deal with like the subjectivity of annotations that we have. We wanted to really combine the different data sources, do multimodal data analysis, so text and images and how it really complements each other, and a little bit about interpretability and explainability. We've already heard a lot about that earlier today, but I will have a few things about what we have done in that domain. XR-Mode is an EU-funded project, so we have 7 partners, academic, industry, and hospitals that are in the project. And it was really meant to be like an application-oriented project. So we wanted to have, we have 2 academic partners, University of Padua and HCSSO. So we worked on text and images, initially separately, and then we combined that further. Same thing for the industrial partners with companies that were supposed to pick up everything we developed on the academic side, certify it, and make it accessible in hospitals. It's not directly our role, but we need to work with them. Then 2 medical partners. And in the back end, we had Surfsara, which is the Dutch supercomputing center. So it's a computing center that allows us to scale things and to really do very large data analysis. And so the objectives of the project are very similar to what I had defined before. So we wanted to train with weak labels, if possibly fully automatic, wanted to look at multimodality, map things onto ontologies, so patches of images onto specific concepts that they would represent. Same thing for text, so we extract the concepts. And also in a multilingual way, because we had histopathology reports from Italy in Italian, we had them in Dutch, we had a few German ones, and then we had a lot of content also in English, so we needed to map that into a domain where it's actually language independent. Make that scalable, and one of the things we wanted to use, because the images are available, is also look into the biomedical literature. It actually models, like much of the knowledge in medicine is stored in the literature. And there's a very large number of images that are available. Not all images, but there's PubMed Central, which is the open access literature that is stored. And they make the data completely available. It's possible to download it via FTPs, I think. 7 million zip files you can download and then unzip for a couple of weeks. But it's a great resource that we wanted to integrate into the clinical data to complement that. We had 4 use cases, so 3 are cancer, colon, lung, and cervix uterus. And then we wanted to have 1 non-oncological case, which is also frequent in histopathology, it's celiac disease. The idea behind that is really to have domains where we have a lot of data and where we can access the data and where we can then check also whether what we will do in one domain, whether we can make it usable in other domains as well. We had a couple from a previous project on prostate cancer as well, and so that's sort of the 5th use case, but that was not directly linked to the project. We started building a platform. We wanted to bring it into the clinical routine, so we had a viewer where we had plugins. We could do annotations, but also show the algorithmic output of what we were developing directly to the histopathologists. It's exactly modeling what they were doing, so they could zoom in and out a bit like Google Maps and like high-resolution images. And then we checked what kind of data we actually had accessible. I mean, difficult to read the details here, but we acquired our own data in the 2 participating hospitals. Then we were looking mainly into big repositories like TCIA, the Cancer Imaging Archive, TCGA, the Cancer Genome Atlas, and taking these data sets out, often with reports, sometimes structured data that we then translated into reports and to sort of build our models. And then we also had a few thousand images from the biomedical literature. Again, with text attached to them, we could do captions or parts of captions that relate to that. And I think it's really important that funding organizations really push for making data available, sharing data, making it accessible. And I think the NIH is really pushing for that, and many national organizations, like the Swiss National Science Foundation as well. And it really helps also other researchers to start more quickly. It's in many AI-related health projects, one of the challenges is getting access to data, and particularly getting started, because there's often an overhead in the beginning to get the data, to have like annotations, labels with that. And that's where I think TCIA, TCGA really help. And I've put up a graphic on the number of the number of articles in the open access literature, number of images scales about it in pretty much the exact same way. But what we can see, it's exponentially growing. There's more open access publications, and these are made available. So we downloaded the last part in 22 with over 20 million images. It's a lot. It's very noisy data, so it's not easy to deal with. Some of these were classified initially as histopathology, and we then retrained the algorithms. I mean, we have compound figures. You can see that on the right side. So we could automatically cut them apart and then look at the subparts. And where we have figure captions that actually associated with one of the subparts, we can directly associate them with that as well. Developed an automatic pipeline, removing like really small images because the quality is not sufficient. Classify them into figure types, so remove everything that we don't want. So here we were really concentrating on histopathology images, use the compound figures, and then look for human versus animal tissue. And there's actually structured data that are available for PubMed Central. So in general, we have the organ, we have the species, also like children, elderly. I mean, a variety of things are made available, manually annotated, so we could also use that, partly also for diseases. Other things we did directly from the images. So here as well, we use text and visual information in this automatic pipeline. I mean, we had mentioned that before, clinical text is not easy to analyze, so it's many non-standard abbreviations. I have a few standard ones here, but even for AB, ABC, you have like seven or eight different ones. There are spelling mistakes in clinical text. Often sometimes they're quickly written. Technical language, nested phrases, double negations, also like little evidence of, I mean, is that negative? Is it a negation or not? There's different levels of that, and obviously synonyms, etc. So that's also why we wanted to go towards something where we would have an ontology for our domains. So we manually created this ontology. We took a subset of UMLs and included more terms that were needed. I mean, there are many advantages that the literature images have. Actually, usually when we have clinical data, there's a distribution where a few cases are very frequent, and then very many cases are very rare, occur like only occasionally. But these are the images usually that are oversampled in the biomedical literature, and that's why we wanted to include them. We couldn't learn only from the literature images. It's too diverse and the quality is not good enough. But here we get the unusual, the untypical images. We have images from many laboratories, many hospitals, so many scanners. Again, we were looking into generalizability much more than pure performance because we wanted it to work out. And again, it's increasingly, exponentially increasing, and particularly when there are new machines, like there's a new CT or MRI scanner with new images that you need your algorithm to adapt to, that's usually the first ones that are published with, because if you have a new technique, that's usually what you would publish on, because it's much easier to get it accepted as well. And, but then obviously we don't have metadata, so clinical, we have a DICOM header, we know the size of pixels, we have slice thickness, or for histopathology, we know the dimensions, the magnification. But again, we can actually develop this as well. So for histopathology, we looked at like a tool to, we generated from the images we had, training data, and then checked it out. Because actually scale, for example, in histopathology plays an important role. If you have like enlarged nuclei, for example, it's characteristic for diseases. So we need to know what is the scale of the images if we want to have very good results. And as I said, so the objective of our work was really to look into the weakly supervised learning. So the first model we developed, we had some strong annotations, so pixel-level annotations in microtissue arrays and so smaller sets of images that we used to then train a teacher model that would then use that on a large amount of non-annotated or weakly annotated data, where we trained a student model, and this was then used to on our test set. And we had different setups to see like what is the influence on what type of data. And we can clearly see, I mean, the more data we can use, also the non-annotated or the pseudo-labeled data, we actually increase the results very clearly in that respect. And then we took this technology basically one step further and really developed an automatic pipeline. So the only manual step here is that we developed the ontology. So for each of our 4 disease categories, we developed this ontology that was developed by a group here in Padova. And that was then, so we knew what kind of labels we would like to extract, we would like to learn from the images, and we could then automatically extract that from the reports, so the weak labels. We would take the whole slide images, we would cut them into small patches, and then we could actually, because we had a sufficiently high number of these images, we could really train them in this way in a fully automatic fashion. And we did that, first of all, in an article that's published in the NPJ Digital Medicine, where we did that on colon images, and then we applied it also for lung cancer, for uterus cervix cancer, and then also for celiac disease. And like that, we can really show that we could really remove the manual annotator and massively remove the effort once we have this domain ontology, and we can easily deploy it on other domains of that if we have a sufficiently large number of annotated samples. And as I said, for this we used the publicly available data set in English, the reports in Dutch and Italian in this case. So we can really show, even using automatic translation, actually works reasonably well when it's a question of extracting specific concepts. So the translation might not read very well, but that's not what we're interested in. We're really interested in extracting the concepts. And so this is basically what I'd shown in the beginning, like on the first slide. So we can really show that we can automate this process, generate the labels once we really understand the domain in a semantic way, map the data, and create the models with a very limited effort. And I think that was the objective of the project that we then reached in the end. And we're still in the process of publishing the last papers on like lung, uterus cervix, and also celiac disease. And so we did a couple of user studies as well in the project, and they are also in the process of currently being published. And one of the things that I also wanted to talk about that's not directly part of the project, but that also came out of it, is that we need interpretability as soon as we want to have user tests, as soon as we want to include it. Because for a clinician, just getting an output of a black box it's not sufficient. They need to integrate it with many other data types. And when you're facing a patient, you need to be able to explain why you come to a specific solution. And I mean, there were a couple of presentations on that just before as well. We actually we organized a workshop where we looked at all of these different terminologies that exist for interpretability, explainability, transparency, accountability, fairness, bias, etc. They're all related, and they're actually used in different ways by different persons, by different groups. So we actually took a workshop where we invited social scientists, philosophers, people from the legal domain, because they're also, with the GDPR, you have a right to have an explanation when your data are used for AI-based decisions. So, I mean, there's a whole definition on that we need to take into account, and I have one graphic from that where we really checked, like, what are the concepts that are used, what is the terminologies that are used, and we try to sort of combine these different wordings, create the links and how they were used in different domains to also make it better understandable. And I had a PhD student who worked on this explainability, and she came up with what she called regression concept vectors. So we actually take existing features, so what clinicians use. So for histopathology, for example, nuclei size is one abnormality. Internal heterogeneity of the cells is, border heterogeneity is. We took these, and we then checked whenever we made a decision for a specific grading staging, in this case on prostate cancer, what exactly, how much of the decision we could actually map onto these specific features that we had, which then allows us to give this feedback and say like, oh, the decision was taken because 20% due to that, 15%, 30% to that. We also, in some cases, had only a partial explanations for what we have. But this is exactly, I think, where we could then go further and say like, okay, what does the algorithm see that we maybe don't see as humans, or what are other features that we could have, come up with hypotheses and work on these kind of things. And when we better understand how the internal part of a deep neural network works, then we can actually also modify it. And we use that in one of our example applications where we actually checked for scale invariance because we pre-trained with ImageNet. ImageNet, I mean, has objects like birds, cars, dogs, cats, and it really wants to be scale invariant, which we really don't want to have. So we checked that out and identified which part of the network are responsible for scale invariance. We deactivated them and had a massive increase in performance on the specific applications in histopathology. So I think if we understand the networks, we can actually also do like manual works to increase the performance. Because all the knowledge we have, I mean, we can feed it back into a system. And I think that's what we need to take, particularly when we have limited amounts of training data. I think it's quite important to actually work on these aspects. And again, I mean, I really want to highlight also for all of the people who work on AI-based application in health, I mean, things work easily in laboratory on a closed data set, but when you have regularly changing data, when you have clinical data, when you have noisy data, we really need to pay attention to what is actually within the distribution of the algorithm, where it can make a decision, what is not. It's also important to actually check what is the impact of a system that we have. How can we make better decisions? Do we have like more confidence in our decisions? Or is it faster? I mean, quite often it's not, particularly in the beginning when you develop a tool. It often slows down clinicians. User tests are complex to set up, but at least in our experience, it really gives a lot of feedback, and we really learn all these small things that can make somebody slower, or that might actually be contrary to clinical like workflow, and really doing an integration with the clinical image viewers, for example. I think that's really important. And whenever we did that, I mean, we really learned a lot from all these user tests that we were doing. And we're now working on the next steps. So we have a new project called Hereditary. It's coordinated actually here in Padova, where we pick up some of the technologies, but we'll actually go much more multimodal. So we use like histopathology data, ophthalmology data, radiology data, different types of images, trying to complement them, signals, text, structured data, genomic, the microbiome, to really work on neurodegenerative diseases like multiple sclerosis, ALS, Parkinson's. So these are the targets. And also looking at the gut-brain links, so what are actually the links between the microbiome and specific diseases or development of the diseases. And like also mentioned by some partners, we're having a federated learning framework that we have developed for this, and that we're currently setting up actually with our clinical partners. And conclusions, so I think interpretability of deep learning is really key for bringing it into clinical workflows. We need to look at like what is really needed at what moment to bring the right information to the right person in the right format. We need to look at like scalability. We can use internal or external resources. We need to look at like how we limit the manual interaction, like to use weak labels wherever we can, and also how to integrate all the data we have. Because clinicians have a lot of information on a patient. When they see a patient, they see what is the status, is the patient like in a good physical shape or not? What is maybe family history? All these kind of things we can integrate in the end. And I mean, all of this is obviously group work. The XML project, we have several other partners who did much of the work that I presented here. This is a picture of our team in Sierre. And you can find more information also on our web pages. So thank you very much.
Thank you very much for the very interesting talk. Questions from the audience? I can start with one. Very interesting. I like the explainability part where you mentioned that you're using some reference features, right, to guide the explanations. So could we see those or think of those as like a linear regression model where you're actually choosing those features that you want to project on, right?
Yes, I mean, there's been work on like concept detection vectors, like really looking at binary concepts, detecting them from images like, I don't know, zebra likeness. I mean, these are, and based on that work, we basically looked into, because much of what we need to do is rather regression task, not binary detection task. So we're looking into, like, I mean, if you have enlarged cells, you have, it's a continuum. And if you have like heterogeneity, it's the same thing. It's a continuum that we're looking at, and that's why we're sort of adapting it for the regression parts and then looking at what part of the network is actually responsible for that. And if we can mathematically model it, then we can actually check out.
And I presume that could also be extended to, for example, could be using some prototype objects or rules that are guided by the experts, and then you say in order for my explanations.
I mean, there's a large application area. We've mainly employed it for histopathology, but it can easily be employed to many other fields as well. The code is on GitHub, it's available, so if anybody's interested, don't hesitate to also contact us.
Very nice talk. I have a question about your co-training of the report with pathology. That's amazing. So I'm curious, for the report, how do you represent the entities, and also if there are numerical values in it, how do you jointly represent them?
I mean, we developed an ontology for like every disease domain. So the paper I presented, it's an ontology for colon cancer. We looked at like what are the labels we're interested in and what are the concepts that are related to these labels. And that's basically what we're trying to extract directly from the text. So we're not trying to be doing a complete extraction of the text. We're really trying to focus on what we're interested in for the learning task. But again, you can possibly extend it, but that's also why in some cases negation is not the most important part for us, because we're looking at specific concepts that we might have, and these subtle negations or double negations, I mean, if we have a sufficiently large number for the learning, it's not always the most important thing.
Great. Is there any other last question from the audience, perhaps? If not, let's thank the speaker.
Our next speaker is Professor Filippo Lanubel. Lanubel. I'm getting better at Italian. He's a professor of computer science and director of the Department of Informatics at the University of Bari, Italy, where he also leads the Collaborative Development Research Group. And today he's going to talk about machine learning development operations for healthcare.
Thank you. Thanks for inviting. I'm aware that I'm the obstacle before the coffee break, so I take this burden, so I will try not to take too long. I will talk about machine learning, actually as part of artificial intelligence, because most AI applications today use data-driven AI. That is what I will talk about. So I will talk about what models that are built based on examples of input and output, and these models are basically will become components that will be part of an inference service that makes prediction on new input to guess some output. And this is because Hold on. Something. I'm trying to, okay. And so, because models, machine learning models don't live by themselves, but to be used, they need to be part of some systems that we'll call ML-enabled systems, that are software systems that include both non-ML components, that is traditional software components, and machine learning components. And there are users that interact with these systems, and there might be also sensors and actuators. And so the problem is how we build these ML-enabled systems. Actually, right now, data scientists tend to focus on machine learning model building. I mean, feature engineering, model training, model evaluation. But the whole process model actually starts from understanding the requirements, collecting data, cleaning data, labeling the data, and after model building, deploying the model within an overall systems and monitoring performance of these models while in production. And, but since the work of data scientists focus mostly on model building, starting from education, what they usually build are models that live within Jupyter notebooks, single models working on data sets that usually already there. But this is not enough to have an impact in healthcare. Means to get these models to affect the life of patients, they need to be part of systems to be in the hand of medical doctors and caregivers or the patients themselves. And this big challenge, I'll try to decompose in specific challenges. The first specific challenge is having the training process be something that anybody can reproduce and can be auditable. In this slide, I report a study that was made on Jupyter notebooks that are public as public sources. High number of public Jupyter notebooks that are on code hosting platform, and most of them cannot be reproduced. And so who knows if what the performance that are published actually are achieved. And sometimes it's because there are broken links to data sets, other times because there are broken dependencies related to the libraries the code referred to. Another specific challenge is about the expectation that building the model is enough, but actually it's just a small fraction of what you need to put the model into production. This figure was produced 9 years ago by Google engineers that showing that what you see in black is the size of code for machine learning for the products of Google's that do translation and recommendation, but most of the code they have and most of the changes that they need to apply are related to code that is not strictly about machine learning. And you need effort and resources for managing this change. And actually, about the resources, another challenge is that you need teams that are made with people coming from different disciplines, different background. In particular, you have data scientists, you have software engineers. For example, I'm a software engineer, but the education, background, experience of software engineers tend to be different from the background of data scientists. Many data scientists come from physics, mathematics, or statistics, and don't know very much about how to develop software. They are able to make programs, but the programs that they learn to make are programs that are usually you have just one user, themselves, and that don't live for a long time. In software engineering, something is specific of software engineering, that you build software that will be used by many people. There will be many users working at them because of the scale, and also the software is expected to live for a long time. And software that lives for a long time means that will be changed many times. And it's not easy to change software done by somebody else, because you need to understand what has been done. And so it's something for which software engineering has been born. But if you have to include also models as part of the system, you need to talk with data scientists, because at some point, data scientists tend to make exploration with Jupyter notebooks, but at some time it's expected that the code that are in the notebooks should become code that lives in files under version control, and this passage is not well regulated and needs communication. And there is another challenge, is about quality. Software engineering usually, as part important of software engineering, is quality assurance, but usually it's related to quality assurance of code. But for machine learning enabled system, you also have not just code, you have models. So you need to make quality assurance of models. And you have to know that differently from traditional software, there is not a strong concept of correctness, because a machine learning model can be wrong sometime, and hopefully not too frequently. And so you need to measure the performance, and you have to define requirements for that, and you expect also to include the humans in the loop. And so the right question is, if the model useful for a problem, rather than is the model always right in the prediction. But there are also data that you have to assure quality, because of course, if you have more data, you can expect to have better models as a performance, but data needs to be right data. So because if you have noisy data with wrong formats, you will have models that don't achieve good performance. And if you have data that are inaccurate because, for example, labels are not applied in the right way, you will have actually biased models. And so even having more data will not solve the problem. And you need to invest in data quality. And in some way, when building machine learning enabled systems, and we are today in a situation close to a situation of some years ago that was true for software, when software building was becoming, there was ambition of developing important software, but there were projects that were failing because engineers did not know how to build software with multiple people involved and with living for a long time. So in this case, Greedy Butch, there was a tweet some year ago that was reminding that it's like we are again in a new cycle where we need to invent something similar to what was invented for software engineering 20 years ago, to make this system scalable and living for a long time. And this is why there was, in these last years, the term MLOps is born as a new term that literally means machine learning operation. Actually means basically the set of practices and tools to facilitate the creation and evolution of machine learning enabled systems. The root of the name comes from a popular term in software engineering that is DevOps. That was a term became popular for accelerating the deployment of software into production. And the emphasis is on the automation. Today I was, one of the presentation was talking about the shortage of skills in the field. And so that is one way to solve this, is also to accelerate on automation, especially for the automation of the steps that are not covered in traditional machine learning field, especially for model deployment and model monitoring. And what I'm presenting now, very briefly, is something that we started to do as part of the DARE research project that was presented today by Professor Lorenzo Chiari. This is a solution framework that we are developing to be tested by the pilot projects, by the more than 60 pilot projects within the DARE research framework. And this is a pipeline of 6 stages. And in this pipeline, there are some practices and tools for each of this pipeline. And the goal is taking a machine learning model built by a data scientist and make it part of a machine learning systems as a component that can be accessed through an API. So very quickly, the first step is scoping an ML problem and to define all the characteristics, so to have also some kind of documentation and record what are the requirements. And then an important, what I was also mentioned at the beginning of my talk, ensuring the reproducibility of the machine learning pipelines. So if you need, if somebody asks you to repeat the training of the model, you should be able just to push a button. And everything should be repeatable. And even if somebody else wants to do that, should be able. And if somebody wants to see what were all the parameters that you were trying, the algorithms, everything should be recorded so that you can also go back. And there are tools for managing this. And then the step to assure the quality of the machine learning artifacts, specifically the models and data. And for example, there are also today some tools for measuring the carbon consumption of the training of a model. These might become a kind of badges to be, like you put badges outside of a refrigerator, you can have something like that also for a model. And then a step for making the model become an API, that means application programming interface, so that can be even uploaded in the cloud and be queried by some client device. And then using the containerization technology to make easier to move the model across different hosts. There can be even different also cloud providers, so that you are not stuck by conditions of some vendors. And finally, being able to monitor the performance of the models while is actually used, because the model could be good once was trained at the beginning, but after that there can be the data for which you are using that might change, because there can be changes to the population, or there can be some specific event that can make things change. And if the distribution of the data becomes different in production, becomes different from distribution of data you use during training, the prediction will not be any more good. And if you realize on the fly that these performance are decaying, then you will need to retrain with the new data the model. So we are starting to build this solution framework, and we are using at the beginning some of the pilot projects, some within there and even some outside also there, and with different use cases of predicting using classification, models for classification of some illness. And we are also trying to make this make easier, not having something too much effort intensive. We are trying to build a template that will make easier to install the required tools, and also that probably will be customized, and to have even organization of the source code that is how it could be expected, with configuration files for the tools that are already there, that will make easier to adopt the framework. We are also doing teaching of this, because we started by teaching these practices and tools, first with our students, Master of Science students in computer science, and this has been also the university course has been also replicated in Spain at Polytechnic of Catalonia in Barcelona, and now also in Belgium. And of course, this to be used for healthcare, there is not just automation, because there are people need to be involved, specifically healthcare professional, professionals need to be involved in the first steps of the pipeline, model requirements, data collection and data labeling, as well as model evaluation and model monitoring. And so this is something that is going on. And so here I'm summarizing what were the main points of my presentation. And there are our acknowledgement to their research project that is supporting this effort.
We have time for 1 or 2 questions. Anyone from? Yes.
Hi. The last slide of the need to involve healthcare professionals, it's a little more important than just a slide, because I see a low-key battle between medical staff or expertise and computer scientists that try to provide to medical experts these new tools to make new discoveries. So as an expert in pipelines of software development, what is your message to medical experts to gain from this and not to make a battle between being replaced and collaborate?
Yeah, that is really a challenge, because right now, you're true, we are experts about the technology, and now with their research projects, we are starting a collaboration with medical doctors and data science is working with medical doctors. My message will be, don't take this work seriously, because otherwise what you do will never, it will be very difficult that we'll go over the paper, the research paper that you publish. Here the challenge is doing something more than a research paper. But that needs, it's easy to say that to have interdisciplinary teams, but it's really tough, because when you work in different disciplines, different environments, everybody has their own business and can find not enough time, or think that is not, priorities could not be the same. So we are starting, and I can tell you that it's difficult, because we could be much faster, but the biggest obstacles is to doing steps together.
I have one question. So you mentioned the importance of the upstream, like data quality control, labeling, etc. Do you have any recommendations of how to
Again,
the data quality control and
Yes.
labeling part? What is your recommendation of incorporating that piece of pipeline?
Okay, my recommendation is the good news that there are some tools that can be adopted. For example, for the noisy data, there are tools in software, in general, in software engineering, there are tools that are used to make automatic testing. There are also tools that can be used to make automatic testing applied to data, to test that at least the data are in the right format. For the wrong labels, there is something that you can do also. So it's something that there are good there is also enough good stuff from which you can develop joint work. That I think they should be then focused on healthcare as a domain.
Okay, thank you.
Thank you very much. So we have a coffee break. I would kindly ask you to stay within 15 minutes. So go quickly to the coffee station, grab your coffee, and we resume here 10 before 5, okay? 10 before 4, sorry.